Benissimo, bentornati a tutti. Ah, ora partiamo con la parte di presentazioni scientifiche. Switcherò in inglese anche per il beneficio dei nostri ospiti esteri. Okay, so good morning again and welcome uh, to this meeting. Again, I'm Matteo Clerici. I'm from the University of Insubria, from the Department of Science and I Technology. And it's really a great pleasure to start the scientific discussion of this gathering. Here with me we have Alessia Anlevi and Marco Lamperti, and together we are going to give you an overview of the scientific activities that are ongoing within our CLIP, the Como Lake Institute of Photonics. And again, I want to acknowledge and thank the Comitato Nazionale and all the supporters for making this event possible. Okay, let me tell you briefly before what CLIP is, and then we'll go through the research activities that defines it. So, let's see if this works. Okay, yes. So the Como Lake Institute of Photonics was founded at the end of 2024, beginning of 2025, by faculty members of the Department of Science. Gosh, this is going. Yeah, that's really not working. Okay, let's try this. Okay, no. Okay, what I was saying. The CLIP was funded by faculty members within the Department of Science and High Technology working in the field of photonics. And at Insubria University, we have a long-standing tradition of research in photonics that traces back to the pioneering contribution of Professoressa Alessandra Andreoni and Luigi Lugiato. And our research and our labs are located at our campus here in Como. Now, CLIP hosts 9 academics and two affiliated researchers from IFN CNR, on top of a growing number of master students, PhDs, and postdoctoral fellows, making these centers one of the largest of the Department of Science and High Technology. So our ambition is to position CLIP as a reference point for photonics research and innovation, both nationally and internationally. And how to do that? Well, what we can do is to engage with the excellence that is already present in our territory, and both nationally also and internationally. And this is really the reason, the motivation for us to launch this event and to have this meeting all together. Okay, here is an overview of the members of CLIP. As I mentioned, we have permanent staff, postdocs, and students. I currently serve as the director. Alessia Levi, Enrico Brambilla, Luca Nardo, and Franco Prati form the scientific committee. Marco Lamperti is our vice director, and Paolo Di Trapani is our. Yes. Now you know. Okay, but let's move from the people to the research activities that really defines our center. So the main area of expertise of CLIP revolves around quantum science and technology, the study and manipulation of matter, integrated photonics and nanophotonics, biophotonics, and indoor lighting technologies. These activities are uptaken by different research groups that I'm going to introduce you, and later we'll see presentation with content from all the different groups. So we have the Quantum Optics Lab by Allevi and Bondani, the Laboratory of Advanced Fluorescence Spectroscopy by Lamperti and Nardo, the Light Scattering Lab with Ferri, Parola, and Anzini, the Ultrafast Nonlinear Optics Lab by Jederkiewicz, Di Trapani, and myself, the Quantum and Ultrafast Photonic Lab with Caspani, myself, and Jederkiewicz, and the Photonic Modeling Group with Prati and Brambilla. On top of that, we have a spin-off company that has been initiated and led by Paolo Di Trapani, and you will hear about that in the next presentation. Okay, now I hand over to Alessia for the presentation of the first research groups, and then to Marco, and I'll pick up at the end.
Thank you, Matteo, for the introduction. So I want to speak about the Quantum Optics Lab, to which I belong, together with Maria Bondani from CNR and Marco Lamperti, who is a tenure track researcher at the University of Insubria. At the moment, we also have two students, Silvia Cassina, who is going to finish her PhD, and Alex Pozzoli, who is going to graduate, and then he will start his PhD. The distinguishing character of our lab is the intensity regime in which we work. In fact, we operate in the mesoscopic intensity regime. This means that we work with pulsed light and in the condition in which each pulse can contain several photons. To this aim, we use as studio detectors photon number resolving detectors. During the last 20 years, we have explored and used different kinds of photon number resolving detectors that are able to count the number of photons contained in each pulse. And in a few minutes, I will present a specific class that is the class of silicon photomultipliers. But first, let me mention the applications in which we do our work. They are all in the context of quantum information science. In particular, we operate in the field of quantum optical communication, for instance, in the underwater environment, and in a moment I will explain you why. And we also working on quantum state discrimination with classical or non-classical states. We have also started working on continuous variable quantum key distribution schemes. And finally, we have some works related to the field of imaging. And in particular, we explore different ghost imaging schemes exploiting super Poissonian states of light. But first, let me say a few words about the two main ingredients of our work. The first case is given by the generation of quantum states of light. In particular, we typically produce mesoscopic twin beam states of light that are entangled in the number of photons. These are two pictures taken quite recently with the new laser source that we bought, and this is actually a sub-picosecond laser source. So, in collaboration with Matteo, we performed a proper shaping of the pump beam to produce bright twin beam states. And I also want to mention that the two arms of these twin beam states are endowed with a multimode thermal statistics, and this is a resource that we use many times for our protocols. The second characteristics of our work is given by the detection in the mesoscopic intensity regime, and in particular, this is the case of photon number resolving detectors, silicon photomultipliers. Why silicon photomultipliers? They are typically used in the context of particle physics because they are cheap, they are compact, they are small, but actually they can be also useful in the context of quantum information, the quantum optics experiments, because their quantum efficiency is not so low. Okay, of course, it's not like the one of superconducting detectors, but there are models that can reach up to 60%. The model that we used in our experiments can reach the 40% in the green-blue region. And this is good, for instance, for underwater quantum communication, because blue light is the one that is less absorbed by water. We use these kind of detectors both in direct detection schemes, like this, picture where we are trying to measure quantum correlations between signal and idlers of mesoscopic twin beam states. But it's also possible to embed these detectors in more complex schemes, like the one that is presented here, that is the so-called homodyne-like detection schemes, because actually it mimics the typical scheme of optical homodyne detection. So the idea is that we want to obtain information about a signal state, and to do this we mix it with a local oscillator. In our case it is a low intensity local oscillator and not a macroscopic one. And at the two outputs of the interferometer we are using two photon number resolving detectors instead of the typically used thin photodiodes, and we can calculate the photon number difference, but we can also have direct access to the photon number distribution, and this single photon number distribution can be used as well to obtain information about the signal. So twin beam states and silicon photomultipliers are important ingredients to perform many of our activities. Here I just want to mention some of the most recent ones, and I also want to mention the main collaborators. For instance, we have recently implemented a some frequency generation based photon number resolving detector, that is a detector that is able to detect light at telecom wavelength with silicon photomultipliers by using a some frequency generation process that is a nonlinear optical process that converts telecom wavelength to visible wavelengths. And we did this in collaboration with the group of Marco Marangoni in Lecco. And we also used this scheme to start a quantum information protocols. The idea is to use a discrete amplitude modulation of coherent states to perform in principle quantum key distribution. The second activity that I want to mention is the realization of a hybrid discrimination strategy in which we use we simultaneously used two resources. The first resource is given by photon number resolving detectors that allow us to measure, to properly measure the mean number of photons, and this is a classical resource. And we also use a non-classical resource that are our quantum states, the twin beam states, to guarantee the security of the communication protocol. This work has been done in collaboration with Luca Razzoli from University of Pavia, and now we are also want to continue this work in collaboration with Gabriele Cenedese. And finally, the homodyne-like receiver that I presented a few moments ago can be used for constructing a discrimination of discrete and modulated coherent states, and actually we are now working on this topic. We are considering 4 coherent states with the same amplitude but different phases. And this is in collaboration with the group of Stefano Olivares in Milan, and also with Michele Notarnicola that is now in Olomouc, Czech Republic. Second part of the work that we do in the Quantum Optics Lab is devoted to education and outreach. This is mainly done by Maria Boldani, who is involved in many Italian and European projects devoted to education. Especially, she is the creator of the project Italian Quantum Weeks that wants to enhance quantum awareness. So the importance and the role, the leading role of quantum mechanics in general public, especially in high school students. And to this aim, we have realized that, especially in the context of the Italian Quantum Weeks, we have realized several editions of an interactive exhibition whose name is Speaking the Unspeakable. So it's a challenge. And the idea is to try to teach quantum mechanics thanks to this exhibition in which we have videos and experiments and panels that explain the different features of quantum mechanics. Maria is also involved in the use of quantum games, so games based on quantum mechanics rules. Especially, I want to mention Q-Tris, that is the quantum version of the Tris game. And then, just to say other things, we have recently published a book devoted to the Nobel Prize in Physics 2022, awarded to John Clauser, Alena Spee, and Anton Zeilinger. Maria has also realized the violation of Bell inequalities in a high school in Genoa, and now she's going to reproduce the same experiment for high school students of the Como and neighborhood here at the university. And finally, I want to mention the event that I organized last March that was devoted to enhance the importance of the role of women in quantum science. Finally, more devoted to photonics, I just want to mention other activities that we usually do every year at university, like the Luna project, that means La natura della luce nella luce della natura, is a project that wants to explain the different fields in which optics and photonics can find their usefulness. And then we have a summer school optics that in the past was actually an international school held partly in Como and partly in Olomouc, Czech Republic. And finally, the International Day of Light that is on the 16th of May. So every year we organize some events devoted to this day at our university. Okay.
I'd like to say we have plenty of time to discuss about the content during the coffee breaks and the other times, but do ask questions. We have time also to answer some questions during the presentations. Whenever.
Thanks, Matteo. Hello, everybody. I'm Michael Alberti, and let's see if this I can work with this. Yeah, I will present two labs of CLIP, whose activity is very cross-disciplinary. We work with chemists and biologists, and we apply physical techniques. So the first one is a lab of advanced fluorescence spectroscopy, where the expertise is mainly timers, so that's simple of few molecules fluorescence spectroscopy. And our main research activities are basically revolve around the use of timers of spectroscopy for study of materials, in particular drug design. And I will show you an example of this kind of application for photodynamic therapy. And then we have the use of single and few molecules techniques to study the conformational dynamics of non-canonical DNA structures like G quadruplexes and the aggregation dynamics of peptides, especially amyloid peptides. Then we have a couple of facility developments that we go through in the last slide. So let's start with time resolved fluorescence. We use a technique which is called time-correlated single photon counting, where we use a pulse laser to excite the fluorescence inside a sample, and then we use a very fast single photon detector to measure the fluorescence light. And we have a detection chain that, together with the detector, gives us a resolution better than 30 picoseconds. So by making a histogram of the arrival time of photons, we are able to reconstruct basically the intensity profile of the fluorescence emitted by the sample over time. And this is a very powerful technique that can be used in a variety of applications like drug design, cosmetics, nutraceutics, and so on. And so here I have an example which is related to the study working together side by side with chemists to enhance the therapeutic effect of photosensitizer, which are drugs which are photoactivated and are used as called photodynamic therapy. So basically you put this molecule, for example, in this case, on the skin to treat skin cancer, and then you illuminate the region that you need to treat. And in this way, you have a high efficacy on the region that you are interested in and limited side effects. And here is the result. And one of these photosensitizers that we have studied is derived by curcumin, which is one of the main components of curry. The point is that in its natural shape, it is not so. It is a photosensitizer, but it's not so effective. So we worked with chemists to try to modify the molecular structure of this molecule in subtle ways, such that we are able to maximize the lifetime of the excited state. And this corresponds to maximizing the energy that is used to really have a therapeutic effect with respect to other effects of the molecule. For the rest of our measurements, we used a completely different system, which is based on a confocal microscope. And the point here is that we are able with the system to detect the fluorescence light, which is emitted by a very small volume of about 1 femtoliter size inside a macroscopic sample. And if the concentration of the molecule that you want to measure is suitably low, you have inside a very small volume, just a few molecules. So when you measure the fluorescence emitted by these molecules, which usually are biological molecules that we label with a fluorescent dye, you don't have a static intensity trace, but you have a trace which is highly fluctuating over time because of the diffusion of the molecules in and out of this observation volume. And by calculating the autocorrelation function of this time trace, we get something like this, and we can retrieve from this curve a few important information. The first one is the average number of molecules inside the observation volume. And the second one is the diffusion time of the molecule, which directly relates to its size, conformation, and so on. So this is a way to study in real time the change in diffusional motion of molecules. As an example, and this again can be exploited for a variety of applications. For example, the study of binding of different biomolecules, for example, the binding of a substrate to a protein. And as an example of this, we studied a system made by the small vitamin biotin, which binds to the large protein streptavidin. Here we have labeled the biotin. Okay, yeah. This is the diffusion, the correlation curve of biotin, which is characterized by a time of about 70 microseconds. And what we see is that when we add streptavidin, the diffusion time of biotin, which is the one which is labeled with the fluorescent dye, increases about threefold. And so this is a signature of the binding. And we can use this technique to probe in real time the binding of different categories of molecules. We can go one step further with this setup. We can reduce the concentration so much that usually we don't have molecules inside the observation volume. So that we have, from time to time, we have a single molecule passing through. And what happens is that if you look at the trace, you have like just some noise and then some spikes, which are the passage of single molecules. So which is the advantage of this way of working with respect to the previous one? That here you can really distinguish single molecule one at a time. And the duration and intensity of this burst are representative of the characteristic of that very specific molecule. So if you work with a very heterogeneous sample, you can look at the molecules one by one, and you are able to find out the heterogeneity in this sample. Example of this technique, we applied it to the oligomerization of amyloid peptides who are involved, for example, in Alzheimer's and Parkinson's disease. And actually, the point is that we have a protein that aggregates and forms some structures like this, but the most neurotoxic species are the small aggregates that happen at the first part of the aggregation stage. And these are very difficult and impossible to study with conventional technique. And as you can see, they are really very heterogeneous. So our technique is perfect to probe how this polymerization process goes on over time. Finally, we can even do another thing and say, okay, instead of using a single fluorophore to label a single biological molecule, why not using two? So here, black, you see a molecule which is drawn like a strand, for example, a single-stranded molecule. And then you have this one is a green emitting phosphor that maybe absorbs slightly in the blue and emits in the green. And this other one absorbs in the green and emits in the red. So of course you expect that if you shine light on this sample, you will see just green light because you are excited the green phosphor. But what happens is that if the molecule has a conformation such that the green and red dyes are close enough, you have a coherent transfer of energy from this phosphor inside the second one. So actually you see mostly red light out of your sample. So with the same setup of the green light, but now having two detectors, one for green and red fluorescence, we can measure the probability of transfer energy from the green to the red phosphor. And this is strictly related to the distance between the phosphors. So what you do is detect the bursts, then you make a histogram of this probability of energy transfer that we call different efficiency, and you obtain sample dynamics. And this is a very broad distribution, but by analyzing, you can see that by different narrower distribution. Each of them corresponds to different conformation of our molecule. So in this case, we are able to distinguish between multiple conformation of the same molecule. And with this, I come to an end for this lab with the new perspectives. The first one is imaging. We are building a new setup to combine our technique with a microscope so that we can have imaging and our techniques at the same time. And then we want also to complement fluorescence-based imaging and fluorescence-based techniques with label-free spectroscopy, in particular Raman spectroscopy, that can give complementary information on the environment that we are studying. And now I'd like to switch to the light scattering lab, whose main expertise is light scattering, both dynamic and static, X-ray scattering, and spectroscopy on one side. And the other side, the general expertise of the group is statistical optics, coherent optics, speckle, and so on. And this translates to two main research areas. The first one is the characterization of biopolymers and nanomaterials through light scattering. And the second one is the development of new instrumentation that combines different techniques on the same sample at the same time. And these are two examples of these research areas. The first one is the study of the protein gel, which is a network of proteins which occurs in our body every time we wound ourselves. We have some blood spilling out, and we have blood coagulation, which is basically is driven by the fact that we have a formation of this network of proteins which create a clog so that the blood stream can be stopped. And the group, by using combined light scattering, spectroscopy, so turbidimetry, and X-ray scattering, has been able to unravel the conformation of this very complex biological network, which turned out to have fractal structure with very specific parameters that you can see here. On the other hand, for what concerns the second research direction, the group is active in the study of perovskite nanocrystals. And one of the important points there is to evaluate their stability over time. So the stability of the size of the particles and the behavior of the spectroscopic properties of the particles over time. And if you want to do this with commercial hardware, you have to take your sample and bring it to different systems, which of course is not so convenient if you want to keep your sample in the same temperature, not shake it up because maybe you have sedimentation and so on. So the group has developed a combined system where you can perform a variety of spectroscopic measurements, and at the same time you can have light scattering. So you can measure the spectroscopic properties and you can make sizing of your particles. And this is all for me, and
Okay, very well. Here we come to the ultrafast optics lab that is led by Vitali Demitrievich, also detached from the Institute of Optics and Technology of CNR, and Paolo Di Trapani, also part of this. So the main area of expertise of this group revolves around linear and quantum optics, the spatiotemporal shaping of ultra-short pulses, and the control of light-matter interaction with tailored fields, also under extreme conditions. For three activities that I want to mention, that are this group has related first to the development of the ideal source to drive to photon absorption, and specifically I'm referring to a source of correlated photon pairs, so non-classical light. This is a work that is going on in collaboration with other teams within CLIP and also with Alessandra Gatti, and you will hear talk tomorrow a bit more about it. Another area of research is laser micromachining of transparent materials. And I'm going to show you the detailed slide later with some of the recent results. But mostly we are working on optimizing light matter interaction for applications that, for instance, include drilling, wire, writing, photonics, and light. And the last part is the development of techniques for controlling extreme events using lasers, such as, for instance, dielectric breakdown. So I want to give you a bite of the research ongoing on the end of laser micro machining and microfabrication. And these results made by Ottavio Jederkiewicz recently within the framework of a European project that is focused to aim at developing a photonic sensing platform design. In this case, the work pertains the fabrication of microfabrication of components in diamond, and this includes micro holes, micro channels, and micro spiller for microfluidic applications, and also micro electrodes that can then travel to quantum meters like centers in diamond. Just a couple of examples here. These four images here show the result of laser microfabrication of diamond using vessel beams. And what is relevant to note is the ability to control all the surface to enhance the interaction with biological compounds of diamond. But what you are seeing microelectron within diamond. So this is generated using a vessel beam. Yeah. Thank you. So what you were here is the growth of a graphitic microelectron that was growing from the bottom side to the upper side of the diamond sample. Which is quite good. Then you use this microelectrons to, for instance, and centers within the diamond, so to create an all-optical and electronical platform within the diamond. Okay, now we move to the quantum and ultra-fast photonics lab. It is led by Lucia Caspani and myself, and Ottavio Jederkiewicz is also part of the team. And it's a new group at the University of Insubria. Both Lucia and I just relocated. Now it's a couple of years almost, but relocated from abroad. So we brought here our expertise in linear and quantum optics and ultra-fast photonics. So our activities are mostly focused on studies of quantum enhancement linear microscopy. That's a project of Lucia. And actually, I want to advertise that people are wanted to build a team. Also, the generation of complex quantum state of light, also in the photonic platform. And then the studies related to quantum and infrared, quantum and classical infrared optics, and ultra-fast quantum probe spectroscopy. Also, in this case, I want to give you a couple of highlights of research. So the first one relates to quantum enhanced spectroscopy. So this is a research that has been done in collaboration with the University of Glasgow, the University of Loughborough, and the University of Strathclyde, on top of the University. And pertains to terahertz radiation, which is that part of the spectrum between 0.1 and 10 terahertz. Right? And this is very important for applications such as imaging and spectroscopy. So coming from the ultra-fast perspective, it's very nice because you can generate single cycle pulses, and then you measure them directly in time domain with the pump and probe approach, so that you can reconstruct the electric field resolved in time. And that way you do it, for instance, kind of scheme. It's called electro-optical sampling. You have your long wave Terence field overlap to your short probe. You send them together to an electro-optical crystal, and then you measure with a polarimetric assembly the electric field of the Terence field by measuring the phase that it is imparted on the short probe. Now, what is nice is you have direct access to the electric field result in time, so that your spectroscopy can be done directly by Fourier transform. You have access to amplitude and phase in the spectral domain and other time of flight. So why is this important? First, this approach, this time-resolved approach to spectroscopy, are recently demonstrated to have even higher sensitivities than other more standard Fourier transform-based approaches, right? Especially for biological studies. Personally, and this is a bit more the drive for our team, there is also a fundamental aspect that is interesting when you perform measurement directly in the time domain. That relates to the question whether we are able to measure quantum states of light with a direct electric field time resolved measurement. And this question is inserted in a more general field of study that is called subcycle quantum electrodynamics. Also, what is relevant here, we are speaking about Terence field, but this approach has been extended to cover the whole infrared spectrum and is really peeking into the visible. So it's a very broadband and powerful approach. Now, if you want to measure the classical state of light, you need enough sensitivity, so to say. And this is a pump and probe approach, and that means typically just to increasing a tiny bit the energy of your probe, because you need to bring the sensitivity up. But unfortunately, as has been shown by predecessor work, if you do that, you shift from a region where you are shot noise limited to a region where the noise really comes from the nonlinear interaction, right? We have a back action effect. So our proposal was to try to increase the sensitivity rather than increasing the energy of the probe that has this nonlinear noise problem by using quantum metrology. In a way, we want to overcome the shot noise limit using non-classical probes. So a result of this effort that shown here, on this figure, you see the Terence and there are two traces there. One is red and one is blue. So the red one is obtained using a quantum probe, and the blue one is obtained a classical probe. In our case, a quantum probe is a twin beam, so a bright non-classical state that is featured by correlations between photon. Now, if we look at the noise measurement, here you see that the noise is the noise is more or less uniform across the time axis, which means that the main source of noise is not within the Terence field itself, but is really in the probe. Now, if you look a bit more closely at the peak of the Terence field, look at the noise, at the standard deviation of your electric field measurement, what you see is that using a quantum state of light, we have half of the noise of the classical one. That means we have increased the sensitivity by a factor 2 in this case. So that demonstrates that it's possible to use the sensitivity of time-resolved approaches to spectroscopy in a way, but to demonstrate that, you really also have to show what happens to the spectrum. So if you do the calculation right, and that's the transform, what I'm showing in this figure is the spectrum, frequency axis. These are two spectra that are in the classical quantum phase, and one of these are every is part about 2. So that curve represent the quantum advantage, both in amplitude and in phase estimation of your spectrum. So in this way, we have shown that, albeit in a specific implementation, it is possible to use the classical state to enhance the sensitivity of time-resolved approaches, time-resolved. And this work is quite fresh, has been published just a few months. Good. So the second example of research that is ongoing within our group, it pertains to quantum enhancement image. Now, this relates to photon microscopy, for instance, and it has long been thought the suggestion of using entangled photons to enhance the photon processes is not new. The idea is that if you want to perform microscopy and you want to go deeper inside your sample, you need to crank up the intensity of your pump light to stimulate this two photon process, right? Now that can eventually cook your sample or end up with other problems. But if you use photon pairs, entangled photons that are already arriving at the sample location where the nonlinear processing occur at the same time in the same space, you expect to have an enhancement in the cross section because they are already there. You have no longer this quadratic dependence from the flux because the two photons comes at the same time and are in the same space. Now, this was proposed and demonstrated it has been done so since few years, but there was no. So the problem was that it was assumed this idea work only in the very low flux regime until you have really just a photon pair for each absorption event into the nonlinear material. And really that results in signals that are too low for any practical application. So what we want to investigate is a mesoscopic and macroscopic regime. So when when you use bright squeeze states of light to generate these entangled photons, but not only in the photon pair regime arriving in your sample. And what we were able to do, thanks to the theory developed by Alessandra Gatti, I suggest you to listen to the presentation that is tomorrow about this. Thanks to the theory and also thanks to our experiment, we have been able to demonstrate that this limit of a single photon pair per absorption event is not really a limit for quantum enhancement. Quantum enhancement remains there up to a factor 10 times larger. Now, this is the first demonstration. Here you see in the graph shown the signal obtained with the quantum state, the blue one, and with the classical state of equal intensity, equal energy, equal beam profile in pulse duration. And you see that the crossover point is not really a 1 photon per mode, actually, but it's 10 times larger. So also these results have been recently published, and I also want to invite you to have a look at the poster of Thomas Dickinson tomorrow, where more details will be provided. Okay, so finally, last but not least, we have the photonic modeling group that here is represented by Enrico Brambilla. So the main research activity of Enrico relates to the modeling of the numerical modeling of high-dimensional entangled and twin beam photon states, especially for what concerns their generation and propagation. And one work that we are doing together in collaboration with Alessandra Gatti and the other experimental groups that I just mentioned is the assessment of the PDC, the ideal parametric down conversion source for stimulating these nonlinear two-photon processes. Well, with this, really, we have concluded the presentations of the main research activities that are ongoing within our center. So I hope you found this presentation useful and stimulating for further discussion. And in this case, I invite you to reach out to all the different persons that really drive each and every one of these research.